CSIR Evolution Chapter 7. Chapter 7, Phylogeny. Phylogeny can be described as the relationship between all the organisms on Earth that have descended from a common ancestor, whether they are extinct or extant. The evidence for phylogeny comes from paleontology, comparative anatomy, and DNA sequence analysis. Phylogenetics is the science of studying the evolutionary relatedness among biological groups, and a phylogenetic tree is used to graphically represent this evolutionary relation related to the species of interest. The main product of phylogenetics is a phylogenetic tree or tree of life. 7.1 Phylogenetic tree. A phylogenetic tree is a way of portraying that history. In the phylogenetic tree, a lineage is represented as a branching tree, where each node, or split, represents a speciation event. Thus, the order of branches of the tree reflects into the order in which lineages are hypothesized to have split. Evolutionary changes in the traits of organisms must be considered in order to reconstruct a phylogenetic tree. Darwin's Fundamental Idea of Descent with Modifications Evolutionary Guide about the evolutionary changes, as it states that all species are descended from a common ancestor. A phylogenetic tree may portray the evolution of all life forms, of a major evolutionary lineage such as the insects, or of a small group of organisms, such as the snail genus Oncomelania, figure 57. It is a common practice to draw phylogenetic trees with the earliest times from the bottom of the tree, while the latest times being at the top of the tree. The trees represented in this chapter show time flowing from left, earliest, to right, most recent. The relative timings of separations between lineages of organisms can also be represented when combined with other data. We do not date those separations. Some of terms used in phylogenetic tree are root, origin of evolution; leaves, current organisms, species, or genomic sequence; branches, relationship between organisms, species, or genomic sequence; branch length, evolutionary time. In cladogram, it doesn't represent time. 7.2 Homology and homoplasy. According to the Darwin's concept of descent with modifications, species that share a recent common ancestor are likely to be very similar. Such recently diverged species share many ancestral traits, which they have inherited from their common ancestor. The traits which were present in the very earlier common ancestor are shared by all of its descendants. Traits that appeared in the most recent ancestor for the first time are acquired by very few descendants. But in all cases, the sharing of traits by a group of species indicates that they are likely to be descendants of a common ancestor. The characters which are obtained from common ancestors and are present in two different species which are closely or distantly related. Such characters are called as homologous traits. These traits may govern certain anatomical structures, behavioral patterns, or DNA sequences. Traits that are shared by most or all of the organisms in a lineage of interest are likely to have been inherited relatively unchanged from an ancestor that lived very long ago. 4. Example: the vertebral column is considered to be homologous in case of all living vertebrates because all the living vertebrates have a vertebral column. All known fossil vertebrates had a vertebral column, and all vertebrates are descended from the same common ancestor. Therefore, the vertebral column is judged to be homologous in all vertebrates. A character that has been derived from a common ancestor but varies structurally as well as functionally is called as derived trait. Though not easier due to complex evolutionary patterns, but systematists have to study the state of the trait in an ancestor and then determine how it has been modified in the descendants in order to determine how traits have changed during evolution. The complexity of evolution is mostly due to two processes: convergent evolution and evolutionary reversals, that usually generate difficulties. There are certain traits that are independently evolved but are subjected to similar selective pressures during evolutionary time scale. therefore become superficially similar as a result of convergent evolution. For example, although the bones of the wings of bats and birds are homologous, having been inherited from a common ancestor, the wings of bats and birds are not homologous because they evolved independently from the forelimbs of different non-flying ancestors. When a character gets evolved with time in descendants and after certain events get reverted back to the ancestral character, such characters are said to follow evolutionary reversal. For example, ancestral frogs had teeth in their lower jaw which are lost in all descendants. Most of modern frogs do not have teeth in their lower jaw, but one frog genus, Amphignathodon, has re-evolved teeth in the lower jaw. These two processes, convergent evolution and evolutionary reversals, generate similar traits which are similar not due to inheritance form a common ancestor but due to some other reasons. Such similar traits are called homoplastic traits or homoplacies. A particular trait may be ancestral or derived depending on the group of interest. For example, unlike dogs or other mammals, rats and mice, both being rodents, have long continuously growing incisor teeth. The character of continuously growing incisors was developed when the common ancestor of rats and mice got diverged from the common ancestors that lead to dogs and other mammals. How to read a phylogenetic tree. A phylogenetic tree displays the order in which lineages split. This example shows the phylogeny of Oncomelanese snails, the intermediate hosts of the human parasite schistosoma. Acquisition of the ability to transmit schistosoma occurred twice during the evolution of this lineage. Figure 57, a phylogenetic tree. Therefore, no other mammals have developed the continuously growing incisors. If one has to reconstruct the phylogeny of a group of rodents, then he must consider continuously growing incisors as an ancestral trait because all rodents have them whereas other mammals lack them. If one has to reconstruct the phylogeny of all mammals, then the continuously growing incisors would be trait-derived in the rodents. This dilemma makes clear that it is important to distinguish between ancestral and derived traits while reconstructing phylogenetic trees. Identification of ancestral traits. One may find the difference between derived traits and ancestral traits difficult to identify because traits often become so dissimilar that ancestral states cannot be recognized. For example, in case of plants, their leaves can form many different structures such as protective spines, tendrils, and brightly colored structures that attract pollinators are all modified leaves. These structures are homologs of one another even though they do not resemble one another closely. To distinguish ancestral traits from derived traits, one method is to assume that an ancestral trait should be found not only among the species of the in-group, the lineage of interest, but also in outgroups. Lineages which are closely related to the in-group but branches off from the in-group before its base on the evolutionary tree is called as outgroup. Traits found only within the in-group, on the other hand, are likely to be derived traits. 7.3 Steps in reconstructing phylogenies. First of all, the group of organisms whose phylogeny is to be reconstructed must be considered, i.e., the in-group and an appropriate outgroup must be considered. The next step is to choose the characters that will be used in the analysis and to identify the possible forms of those characters, states or traits. A trait stands for presence or absence of a character, or one of the states a particular character may have, such as the number of body segments or number of appendages. To distinguish whether the character is ancestral or derived is the next step and is usually the most difficult step. Finally, through phylogenetic analysis, systematists must distinguish homologous from homoplastic traits. Because organisms differ in many ways, systematists use many characters to reconstruct phylogenies. Morphological characters can be preserved in fossils, whereas Molecular characters are rarely preserved in fossils, behavioral characters are not at all preserved. Systematists use physiological, behavioral, molecular, and structural characters which can be assessed in both living and fossil organisms. Phylogenetic trees reflect evolutionary patterns in more extents as more extensively the characters are studied. 7.3.1 Use of morphological and developmental traits. Morphology gives an important source of information for systematists, that is, the sizes and shapes of body parts. A large amount of morphological data has been collected since organisms are being studied for centuries, and organisms are being preserved extensively in museums and herbariums whose traits can be measured. Systematists are able to measure and analyze the structures of much finer organisms due to availability of wide range of advanced technological tools, such as the electron microscope and computer simulations. Even the molecular level studies are possible due to such advances. Many organisms share similar early developmental stages, but generally, these similarities are lost at later developmental stages. For example, the larvae of marine creatures called sea squirts have a rod in the back, the notochord that disappears as they develop into adults. All vertebrate animals also have a notochord at some time during their development, figure 58. Due to this similarity in developmental stages, one can believe that sea squirts are more closely related to vertebrates than would be suspected if only adult sea squirts were examined. A larva reveals evolutionary relationship. Sea squirt larvae, but not adults, have a well-developed notochord that reveals their evolutionary relationship to vertebrates, all of which have a notochord at some time during their life, sometime during life cycle. In adult vertebrates, the vertebral column replaces the notochord as the support structure. Figure 58, a larva reveals evolutionary relationship. The idea about where and when the organisms once lived and what they looked like is easily obtained from studies of the fossils. Fossils give important clues, which help us to distinguish between ancestral traits from the derived traits. Even the time of divergence of two lineages and their independent evolutionary history can also be obtained from fossil records. But, for some groups of organisms, a very few or no fossils records are found. Thus, the evolutionary history cannot be read directly from fossil record because fossil can only set a minimum age for a lineage. Fossils can give sure information about when the organism was present, but it cannot state that whether the organism was present prior to the event of fossilization or not. Because there is a possibility that the organisms might be present before the fossilization time, but their fossils are not identified or observed. Therefore, the authenticity of phylogenetic trees obtained from fossil records has a question mark. More reliable methods based on molecular data are available now today, which are used to reconstruct phylogenies. For instance, the earliest fossils of liverworts are found in late Devonian, of rhinoceros in early Cretaceous, and vascular plants in the later Silurian/early Devonian. Anthoceros, hornworts, fossils have not been recovered. From the fossil records, one would say that the vascular plants appeared first and then liverworts, whereas the phylogenetic studies based on molecular data suggests that liverworts evolved first and then vascular plants. Figure 59. Figure 59. Fossil-based phylogeny and its comparison with molecular phylogeny. 7.3.2 Use of molecular for reconstructing phylogenies. As time passes, the molecules constituting organisms also diverge among lineages as they are inheritable traits. Primary structures of proteins and nucleic acids, i.e., DNA and RNA, are commonly used molecular traits in construction of phylogenies. 1. Protein primary structure. By comparing primary structure of proteins, relatively precise information about phylogenies can be obtained by comparison of the primary structures of proteins. Two lineages can be compared for genetic differences by obtaining homologous proteins from both of them and determining the number of amino acids that have changed since the lineages diverged from a common ancestor. 2. DNA base sequences. An excellent evidence of evolutionary relationships among organisms can be obtained from the base sequences of DNA. Eukaryotic cells have genes in their mitochondria as well as in their nuclei. Additional chloroplast genes are present in plants. The chloroplast genome, cpDNA, which is used extensively in phylogenetic studies of plants, has changed little over evolutionarily time. Mitochondrial DNA, mtDNA, has been used extensively for studies of evolutionary relationships among animals. By sequencing more than 10,000 base pairs which make a segment of DNA that includes a hemoglobin pseudogene, a non-functional DNA sequence derived early in primate evolution by duplication of a hemoglobin gene, scientists investigated the relationships among the apes. The outgroups for the analysis were ateles, the spider, monkeys of tropical America, and macaca, the rhesus monkey of Asia. According to the DNA data, which is also supported by other types of molecular data, chimpanzees and humans have a more recent ancestral history than they do with gorillas. 7.4 Reconstructing a simple phylogeny. Let us take the example of 8 vertebrate animals such as the lamprey, perch, pigeon, chimpanzee, salamander, lizard, mouse, and crocodile, in order to explain the construction of phylogeny. At initial stage, we will assume that a given derived trait evolved only once during the evolution of these animals, and that no derived traits were lost from any of the descendant groups, Table 10. To simplify the situation, we will select traits that are either present, plus, or absent. It is observed that a group of jawless fishes, called the lampreys, is thought to have separated from the lineage leading to the other vertebrates before the jaw arose. Therefore, we will choose the lamprey as the outgroup for our analysis. Derived traits are those that have been acquired by other members of the lineage since they separated from the lamprey. We can observe that there are 2 unique characters in chimpanzee and mouse, i.e., mammary glands and fur. Those traits are absent in both the outgroup and the other species of the ingroup. Therefore, we infer that mammary glands and fur are derived traits that evolved in a common ancestor of chimpanzees and mice after that lineage separated from the ones leading to the other vertebrates. In other words, we provisionally assume that mammary glands and fur evolved only once among the animals in our ingroup. The pigeon has one unique trait, feathers. As before, we provisionally assume that feathers evolved only once, after the lineage leading to birds separated from that leading to the mouse, chimpanzee, and crocodile. By the same reasoning, we assume that keratinous scales evolved only once, after the lineage leading to crocodiles, birds, and lizards separated from the lineage leading to mammals. We assume that claws or nails evolved only once, after the lineage leading to salamanders separated from the lineage leading to those animals that have claws or nails. We make the same assumption for lungs and jaws, continuing to minimize the number of evolutionary events needed to produce the patterns of shared traits among these 8 animals. From the above information we can reconstruct a provisional phylogeny, Figure 60. Here we can assume that animals which share common unique derived traits have a common ancestor. This common ancestor is not shared by other organisms which do not have the character in question. For example, that mice and chimpanzees, the only 2 animals that share fur and mammary glands, share a more recent common ancestor with each other than they do with birds and crocodiles. Otherwise, we would make unnecessary assumption that the ancestors of birds and crocodiles also had fur and mammary glands. The possible phylogeny of these 8 vertebrates based on the traits we used and the assumption that each derived trait evolved only once is shown in Figure 61. This particular phylogeny was easy to construct because the animals and traits fulfilled the assumptions that derived traits appeared only once in the lineage and that they were never lost after they appeared. Our second assumption on constructing the phylogeny would have been violated if we would consider snake in the group. Because, the ancestors of snakes, the lizards had limbs that were subsequently lost along with their claws, in snakes. Some additional traits are needed to be examined in order to determine that the lineage leading to snakes separated from the one leading to lizards long after the lineage leading to lizards separated from the others. The analysis of a number of traits shows burrowing lizards eventually became adapted to a subterranean existence and snakes were evolved. Figure 60, a probable phylogeny of 8 vertebrates. This phylogeny was constructed from the information given in Table 6 using the parsimony principle. Except for the lamprey, all of the animals displays at least one derived trait. 12.5 Principle of parsimony and maximum likelihood. In case of vast majority of cases, the reconstruction of phylogeny does not work by the simple method we use to reconstruct the vertebrate phylogeny, because fossil and other evidences suggest that traits can change more than once or undergo evolutionary reversal. Several methods are used to deal with these complexities. The most widely used is the law of parsimony. The law of parsimony simply states that one should prefer the simplest hypothesis that is capable of explaining the observed data. Its application to the reconstruction of phylogenies means minimizing the number of evolutionary changes that need to be assumed overall characters in all groups in the tree. It can also be said that the best hypothesis is based on the fewest homoplaces. The law of parsimony is most appropriate to use not because the evolutionary events occurred likewise, but because it is generally wiser to adopt the simplest explanation that can account for the observed data. Complicated data are accepted only when the evidence requires them. Phylogenetic trees are hypotheses about evolutionary relationships and are continually updated with the discovery of new fossil evidences or as additional traits are measured. The maximum likelihood method is another method of reconstructing phylogenies, and is used primarily with reference to molecular data. The computer programs employed in this method are complex as they are designed to deal with the fact that mutations commonly change nucleotide sequences. Reconstructing the phylogeny for any group of organisms is quite difficult irrespective of the method used. For example, there are 34,459,425 possible phylogenetic trees for a lineage with only 11 species. Computer programs using the parsimony principle employ various search routines that calculate the shortest phylogenetic tree, the one with the fewest homoplaces for a given data set, and then compare other possible phylogenies with the shortest one. If, as is usually the case, several trees are of equal minimum length, they can be merged into a consensus tree that retains only those lineage splits that are found in all the shortest trees. In a consensus tree, groups whose relationships differ among the trees form nodes with more than 2 branches. These nodes are considered unresolved because during a speciation event, a lineage typically splits into only 2 daughter species. 7.6 Evolutionary relationship in biological classification. Carolus Linnaeus, a Swedish biologist, developed the biological system of classification which has been used since 1758. This system is based on binomial nomenclature and allows scientists throughout the world to refer unambiguously to the same organisms by the same names. Linnaeus gave each species 2 names, one identifying the species itself and the other the genus to which it belongs. A genus, plural, genera, adjectival form, generic, is a group of closely related species. In many cases, the name of the taxonomist who first Proposed the species name is added at the end. Thus, Homo sapiens linnaeus is the name of the modern human species where Homo is the genus to which the species belongs, and sapiens identifies the particular species in the genus Homo. Linnaeus proposed the species name Homo sapiens. You can think of the generic name Homo as equivalent to your surname and the specific name sapiens as equivalent to your first name. The generic name is always capitalized, whereas the name identifying the species always lowercased. Common names of organisms are not given in italics, but scientific names are always italicized, for ease of writing. When a generic name is written, it is spelled completely for the first time and subsequently the genus name is given in the form of initials, for example, Drosophila melanogaster written completely for the first time, and then followed by D. melanogaster for subsequent times wherever it comes. Recognizing and interpreting similarities and differences among organisms is easier if the organisms are classified into groups that are ordered and ranked. When a group of organisms is treated as a unit in a biological classification system, then it is called as a taxon, plural, taxa, for example, the genus Oncomelania. In the Linnaean system, species and genera are further grouped into a hierarchical system of higher taxonomic categories. The taxon above. The genus in the Linnaean system is the family. The names of animal families end in the suffix idea and are based on the name of a member genus. For example, the family name Formicidae is based on the genus Formica, which include all types of ants, whereas the family Hominidae is based on Homo, which contains humans and our recent fossil relatives, as well as chimpanzees and gorillas. Same procedures are involved in case of plant classification, except that the suffix ace is used with family names instead of id. Thus, Rosaceae is the family that includes the genus of roses, Rosa, and its close relatives. Many families together form orders, many orders together form classes, and classes in turn form phyla, singular, phylum. Finally, different phyla unite together to form the kingdoms such as Plantae, Fungi, and Animalia. Biological classification systems and the unique names improve our abilities to infer relationships among organisms. Beside this, these names and systems have a great significance for several reasons, such as they provide memory and precise communication. Without grouping the organisms, it is like next to impossible to remember the characteristics of many different organisms just based on shared characters. Classification systems are also useful as predictors. For example, the discovery of biochemical precursors of the drug cortisone in certain yams of the genus Dioscorea stimulated a successful search for higher concentrations of the drug in other Dioscorea sp. 7.6.1 Current biological classifications reflect evolutionary relationships. The design of biological system directly related with relationships among organisms. The features that are used to classify the organisms are affected by the kind of relationship we wish to express. Suppose we are concerned about a system that would help us decide what plants and animals were desirable as food. For instance, we might devise a classification based on tastiness, ease of capture, and the type of edible parts each organism possessed. Early Hindu classifications of plants were designed according to these criteria. Although such classification systems served the needs of the people who developed them, but today's biologists do not use such systems now. Most taxonomists today believe that biological classification systems should reflect the evolutionary relationships of organisms, and that taxonomic groups should be monophyletic. A clade or monophyletic group contains all the descendants of a particular ancestor and no other organisms. Polyphyletic groups are those containing some members that do not share the same common ancestor. Paraphyletic group contains some, but not all, of the descendants of a particular ancestor. Figure 61, Taxonomic groups, monophyletic, paraphyletic and polyphyletic. Taxonomists believe that polyphyletic trees are not appropriate taxonomic units, therefore a single cut in the polyphyletic tree can remove a monophyletic group as demonstrated in Figure 58. Since many organisms have not been studied well enough to distinguish characters that are homoplaces, thus the classifications used today still contain many polyphyletic groups. In order to eliminate phyletic taxa, taxonomists change their classifications as soon as they detect such homoplaces. Not all taxonomists prefer strict phylogenetic classifications, but some believe that classification systems should also reflect degrees of difference among organisms. According to this view, certain paraphyletic groups that have undergone rapid evolutionary change and diversification and evolved distinctive characters should be retained. Such groups are called grades. This perspective can be illustrated using birds, crocodiles, and their relatives. Traditionally, crocodilians are grouped with snakes, lizards, and turtles in the class Reptilia, while birds are grouped in a separate class called Aves. Figure 59b. Interestingly, some molecular evidences are available which suggest that birds and crocodilians, a group that includes crocodiles and alligators, share a more recent common ancestor than crocodilians and turtles share with snakes and lizards. Figure 62a. Since the two lineages separated, birds have evolved at much faster rates than that of crocodilians, thus the classification could emerge. Crocodile obtained very few new characters which were different from their ancestors, whereas birds could rapidly evolve significant and distinctive adaptations for flight. As a result, crocodilians are more similar in many ancestral features to snakes and lizards than they are to birds. Therefore, crocodilians look like lizards. Since birds are excluded from the traditional class reptilian, the class gives an example of paraphyletic group because it does not include all the descendants of its common ancestor, that is, birds are excluded. Figure 62b. If only monophyletic taxa were permitted, birds would be grouped with crocodilians in a single taxon separate from snakes and lizards, or birds would be included within an expanded class reptilians. As birds developed unique derived characters which are totally different than their ancestors, they are kept in a separate class. This situation also retains reptiles as a separate paraphyletic group excluding birds. Some of the most familiar taxonomic categories such as gymnosperms and reptiles are paraphyletic groups despite of the preference for classifications which eliminate paraphyletic groups. Because of their familiarity and the extensive literature devoted to them, these categories are likely to remain in use even after their formal taxonomic designations have been changed to better represent their evolutionary relationships. Figure 62, Phylogeny and Classification. A. Evolutionary relationship among mammals, reptiles, and birds. B. The traditional classification unites crocodile and turtles with lizards and snakes in the paraphyletic taxon, Reptilia, which excludes birds. 7.6.2 Use of phylogenetic trees. In order to determine evolutionary relationships among organisms, phylogenies are usually reconstructed while solving and understanding much of the biological problems. Scientists refer the information about these relationships. Many biological statements are really phylogenetic assertions. Any claim of an association between a trait and a group of organisms is actually a statement about when during the history of the lineage the trait first arose and about the maintenance of the trait since its first appearance. For example, the statement that the cytoskeleton is a trait possessed by all eukaryotes is an assertion that the cytoskeleton is an ancestral trait that has been maintained during the subsequent evolution of all surviving eukaryote lineages. In this section, we will illustrate how phylogenetic trees can be used to determine how many times a particular trait may have arisen during evolution, to assess when lineages split, and to explain how evolutionary radiations came about. 7.6.3 How many times has a trait evolved? Generally, flowering plants tend to avoid self-fertilization and reproduce by mating with another individual, and the process is called as outcrossing. Some species have self-compatible individuals that are able to fertilize among themselves. The evolutionary emergence of self-crossing can be verified by plotting the outcrossing and selfing species on a phylogenetic tree. The evolution of fertilization mechanisms was examined in Linanthus, a genus in the phlox family, a lineage of plants with a diversity of breeding systems and pollination mechanisms. The outcrossing, self-incompatible, species of Linanthus have long petals and are pollinated by long-tongued flies. The self-compatible species all have short petals. The investigators reconstructed a phylogeny for 18 species in the genus using a nuclear ribosomal DNA sequence. By artificially pollinating flowers with their own pollen or pollen from other individuals and observing whether viable seeds formed, each species was studied for whether it is self-compatible or reprocessed by cross-fertilization. In case of Linanthus, evidences suggest that self-incompatibility was the ancestral state. 1. Multiple origins of self-incompatibility have not been found in any other flowering plant family. 2. Self-incompatibility depends on physiological mechanisms in both the pollen and the stigma, the female organ on which pollen lands, and requires the presence of at least 3 different alleles. It seems that development of self-incompatibility would be easier than the reverse. Change. 3. Although the sites of pollen rejection vary greatly in other plant families, all self-incompatible species of Linanthus have stigma as the site of pollen rejection. If we assume that self-incompatibility was originally present in ancestors, then the reconstructed phylogeny suggests that self-compatibility has evolved 3 times within this Linanthus lineage. When the self-compatibility was emerged, by the same time evolution of reduced petal size was also occurred. Interestingly, the striking similarity of the flowers in the self-compatible groups led to their being classified as members of a single species. The phylogenetic analysis shows them to be members of 3 distinct lineages. 7.6.4 When did lineages split? The explanation of how phylogenetic analyses can help us determine when lineages split is given by studies of carassiform fishes. The sizes of around 1,200 species of South American freshwater fishes and about 200 African fishes vary greatly in size, shape, and diet. Analyses of mitochondrial DNA and anatomy suggest that the carassiform fishes are monophyletic. When the slowly evolving rRNA genes were studied for sequence similarities, 3 lineages with closely related species on both sides of the Atlantic Ocean were identified. Since all these species live only in fresh water, they were less likely to disperse across the Atlantic Ocean. The genetic differences between the rRNA of the African and the South American species in all 3 lineages are great enough to be consistent with a split caused by the separation of Africa from South America, which is believed to have happened about 90 million years ago.